Good day, everyone, and thank you for joining the Animal Welfare Group Nigeria. It's a pleasure to have you to be part of our meeting today, and I'm so excited about our speaker, Professor Melissa Bateson. But before I go on, I'm Dr. Yasiri Oluwaseun Sarah, um, an associate professor at the Federal University of Agriculture, Abeokuta, Nigeria, and I just finished the Alexander von Humboldt Fellowship in Germany. So I'm happy to welcome you all to MIST. Briefly, I would like to speak about the mission of Animal Welfare Group Nigeria and how it started. For those of you that are joining us for the first time, the Animal Welfare Group Nigeria was started in the year 2019 after the gathering of students and researchers who are interested in animal behavior and welfare. We have three main missions. The first is to increase awareness in animal behavior and welfare. Secondly, is to foster collaboration among researchers who are interested in this area of expertise. And thirdly, to educate the public on the importance of animal behavior and welfare. And over the years, we have been having series of webinars and virtual inaugural lecture. Today marks the 15th virtual inaugural lecture of the Animal Welfare Group Nigeria. So our guest speaker for today is Professor Melissa Bateson. So I would like to take her bio before she goes on with her presentation. Melissa Bateson is a professor of ethology, Biosciences Institute, Newcastle University, United Kingdom. Melissa is a behavioral biologist with a strong track record in evolutionary motivated empirical research on the behavior of human and non-human animals. Her current research focuses on understanding how stressful lifetime experiences and specifically exposure to limited and unpredictable food alter cognition, behavior, and biomarkers of health and well-being. She developed the hypothesis that biomarkers of biological age and specifically telomere length can be used as integrative biomarkers of cumulative stress exposure. She has applied her findings to address problems in animal welfare and human food insecurity. She is committed to increasing the reproducibility of behavioral science through refinement in methodology and adopting open science practices. She is currently the president of the Association for the Study of Animal Behavior. And with that opening, I would also like to add that Melissa Bateson was actually my second supervisor when I was doing my PhD at the Newcastle University. So I'm so excited to see her after 10 years, Melissa. It's after 10 years. So with that, I say thank you so much for honoring the invitation to speak to our group on this interesting topic, which is novel to us, and we believe we'll have a whole lot to learn from your presentation. To the audience, thank you once again. Feel free to introduce yourself in the chat box. Let us know who you are and where you're joining us from. While the presentation is going on, you kindly drop the questions in the chat box, and the questions will be read immediately after her presentation. Thank you so much. Melissa, you have the floor. Thank you.
Thank you, Seun, for a very kind introduction. Okay, so what I want to do today is talk about the use of biomarkers in the assessment of welfare, and specifically talk a little bit about whether telomere length could be a good candidate biomarker. Okay, so to give you an overview of what I'm going to do today, I'm going to start by making some quite general comments about welfare indicators and their reliability and validity and what we mean by these terms. I'm then going to go on and talk more specifically about telomeres as a candidate biomarker that we can use as a welfare indicator. I'll explain what telomeres are and how they relate to welfare. Then I'm going to talk a little bit about my own research. So the research that I've done myself on telomeres has mostly been in the context of understanding effects of limited and unpredictable food in European starlings. So I'll tell you about 3 of the studies that I've done in that area to give you a sense of how you can use telomeres in the assessment of cumulative experience in non-human animals. and I'll finish with a few conclusions about what I think about the value of telomeres. Okay, so animal welfare means different things to different people and is defined in different ways. To make sure we're all on the same wavelength here, I'm going to just start by putting up Meller's 5 domains model, which I think is quite a useful summary of what we mean by animal welfare. And it's one that it's a model that many animal welfare scientists today, I think, subscribe to. So the idea here is that there are a number of physical or functional domains, including the nutrition an animal has, its physical environment, its health, and its ability to perform species-specific behavior. So these domains all affect the mental domain, which reflects the kind of emotional or affective state of the animal. And we feel that this is the kind of central aspect of welfare that we're interested in as scientists. So ultimately, we're interested in whether our animals are happy or unhappy, to use human terms. Okay. So as an ethologist, this is a bit problematic because in ethology, the subjective experience of animals was not regarded as a proper subject for scientific inquiry. So I put up this quote here from Nico Tinbergen, who said that because subjective phenomena cannot be observed objectively in animals, it's idle to either claim or deny their existence. So what are we doing here? How can we do science in this area? There are a lot of problems clearly with inferring mental or affective states in animals. So we have as a central problem the other minds problem. Subjective feelings are private and inaccessible. We only really know our own. I can't even know what yours are like. And it's much harder yet to know what the experiences of a non-human animal are. Maybe you start to have some feelings about primates, but when it comes to something like an octopus, how do we know where to start? Animals' affective states might be very different from ours. We have to acknowledge that fact. We don't know which species or stages of development have affective states. So these are all really big problems. And I think what this means is that we need objectively measurable indicators of affective states if we're going to start to address this problem in non-human species. Focusing on the subjective feelings is problematic. We're lucky in that subjective feelings are only really one component of affective states. So I've just put up hunger here as an example of a particular state that has a strong emotional component to it that we might experience. And what I'm trying to do here is make the point that there are many different components of hunger other than the mental domain, the subjective feelings, which in humans may be accompanied by feelings of fatigue, hunger pains, anxiety, distress, anger, or possibly even shame at not having enough to eat. But there are also physiological components. So there may be changes in blood glucose, appetite hormones, stress hormones. There may be changes in cognition, how much we value food, how much we attend to food, how impulsive we are in our decision making. And there may be changes in behavior. So how much time we spend foraging for food, how fast we eat, our food preferences, how much we're prepared to take risks to get food, and how aggressive we are to access food. So the point here is that physiology, cognition, and behavior can all be measured objectively in animals, whereas we can only infer subjective feelings from measurements of valid indicators. So we have to concentrate on these domains that we can actually objectively measure. Okay. So these reflect a number of different types of welfare indicators that we use in animal welfare science. There are cognitive indicators, and I think I put these at the top because for many people, I think these are now regarded as perhaps the gold standard for assessing affective states in non-human animals. So I'm talking about things here such as judgment bias, or possibly attentional scope, which is a new cognitive measure that's been proposed recently. So we have these kind of cognitive measures that reflect differences in decision making under different affective states. while these might be the gold standard for many people, they're often very difficult to measure. They can take a long time it can take a long time to train animals on the tasks necessary to measure these kind of traits. So they're often not very appropriate in applied situations, such as the assessment of welfare in large numbers of farm animals, for example. Behavior is what we've traditionally used in animal welfare science. So there are many behavioral indicators that might reflect subjective states. So there are things such as posture, which can change with pain, ear position, which might reflect affective states. There's some evidence for that, and other behaviors such as play that might be linked to positive affective states. So behavior can be very useful, but it can also take a long time to measure, especially in large numbers of animals, and its measurement is often somewhat subjective, so it's difficult to measure it reliably in many cases. I've also put physical injuries on my list because these are often used, especially in laboratory and farm animals. So I'm talking about things like bites, bald patches, keel bone fractures that can often be lasting symptoms or marks of behaviours that have taken place in the past. So these can be quite useful, I think, and can be quite quickly assessed in many cases, especially things like bites or bald patches would be fast to measure in, for example, farm animals where you want to assess a lot of animals quickly. The last type of indicator on my list are what I'm calling biomarkers. So these might be things like stress hormones, measures of oxidative stress, and markers of telomere length, which is what I'm going to be talking about today. So what's a biomarker? I would define a biomarker as an objective and quantifiable molecular, histological, radiographic, or physiological trait that are characteristic of biological processes or conditions and that may be used to assess health and well-being. So that's a rather long definition there, but the point is that these are things that we can measure often in tissues such as blood, and that reflect some kind of state in the animal that's characteristic of a particular biological process. So biomarkers can often be very useful because they can be quite quick and straightforward to assess if you have the right tests set up. And they might give you indications of what's gone on in an animal or a human over a prolonged period of time. So I just put up this example here that in humans, the blood level of cotinine, which is a metabolite of nicotine, is actually a very good biomarker of smoking behavior. And it's something that can be measured quite straightforwardly to look at a person's exposure to nicotine, either through smoking themselves or passive smoking over a period of time. So biomarkers, I think, can potentially be very useful as a way of getting at a human or animal's experience, and potentially for looking at whether health or well-being has been impacted in some lasting way by that experience. So if we're going to look for good indicators of affective state in animals, we want to find ones that have various characteristics. They need to be reliable, and by that I mean that they allow repeatable measurements to be made. So we need biomarkers that are going to be repeatable. If we measure them multiple times, we want to get the same results, assuming nothing's changed. We also need them to be valid. They need to measure the right thing, in this case, some kind of indication of the animal's affective state. So both of these characteristics of welfare indicators, reliability and validity, are going to be affected by the accuracy and precision of individual measurements. So this is something that I just want to say a little bit about because it's, I think, incredibly relevant in relation to a lot of biomarkers where there can be issues with measurement error. So if we talk a little bit about accuracy and precision first, these are attributes of repeated measurements of the same true quality. So if I was to take a blood sample with a certain telomere length in it, and I try to measure that, we can characterize accuracy as being to do with whether my measurements fall on center on the true value of the telomere length in that sample. So this bullseye in the bottom right-hand corner here reflects telomere measurements centered around the bullseye, which is the true measure of telomere length. So that would be high accuracy. And this one on the left here shows lower accuracy. So my measurements, my individual measurements, which are the small black dots, are centered around a point which is off center here. So it's biased in this case, a biased measurement. The top row shows very precise measurements. So there's very little variance in the individual measurements. They're highly clustered. And the bottom row shows more variable measurements. So these are quite imprecise. So precision describes the variability of measurements, and accuracy describes how biased they are, whether they're centered on the true value or not. And a point to make here is that both accuracy and precision of measurements will affect both the validity and the reliability of indicators. So we need to really pay attention to how we measure things in order to get meaningful measurements of welfare. So a little bit more about what we mean by reliability. Reliability is about the repeatability of measurements, the extent to which measurements are repeated when a study is done under the same conditions. As I've just suggested, unreliable indicators will often have high measurement error. So both the accuracy and precision with which individual measurements are made affects reliability. Reliability will be higher if measurements are more accurate and more precise. What about validity? Well, validity concerns the extent to which an indicator measures what we're actually wanting to measure. In this case, affective state or subjective feelings. We sometimes refer to this as construct validity. There are different kinds of validity that you'll come across in the literature, and whether we're measuring what we really want to measure is construct validity. And this has started to be questioned quite a bit in animal welfare science, particularly by my great friend and colleague, Georgia Mason, who's been very concerned with how we can establish validity of welfare indicators. Arguably, validity is more important than reliability. It's probably better to measure the right indicator somewhat unreliably than the wrong indicator very reliably. So I think that's a trade-off worth thinking about there, that we want to be sure that we're measuring something that really does reflect affective states in our animals, even if we're doing it not as reliably as we would ideally like. So unlike accuracy, validity is not an intrinsic property of an indicator. It has to be established by comparing the indicator with some kind of gold standard or ground truth. And there are various ways in which we can do this. There are a number of different methods for establishing the validity of welfare indicators, but just to mention a couple of common ones, one thing we can do is explore the association between our candidate indicator and affective state in humans. So, for example, we might do this by looking at whether our candidate indicator is associated with negative affective states such as depression and anxiety in humans, where humans can self-report their affective states. So they're a gold standard for establishing indicators that are associated with affective states. A second approach in animals is to test the response of our indicator to a validated manipulation of welfare in animals. So we might do an experiment where we expose our animals to a manipulation that we have really good reason to believe will alter their welfare in a specific direction, and we can measure how our indicator responds to that experimental manipulation. And I think arguably both of these sources of evidence are really important in establishing the validity of welfare indicators. Valid indicators need to be both sensitive, by which I mean good at detecting cases of negative or positive affective state. And they should avoid what we call false negative errors. So that's where we miss true cases of negative welfare, for example. So we want our measures to be sensitive to positive or negative welfare. We also want them to be specific, and by that we mean sensitive only to affective state and not to other factors. So by this we mean prone to false positive errors. So this would be false alarms. So we don't want an indicator to suggest that an animal is in a negative affective state when in fact it's totally happy and fine. So we'd like our valid indicators to be both sensitive and specific. So just to summarise a bit of what I've said there, I think an ideal welfare indicator would look a bit like this. So if we have the value of our indicator on the y-axis here, going from low to high, this might be from, for example, short telomeres to long telomeres, and we have the x-axis describing the valence of the affective state of an animal, going from negative on the left to positive on the right. A good welfare indicator would have this sort of linear monotonic relationship here, where there's a straight line with a positive slope. So the more negative the affective state of the animal becomes, the lower the value of the welfare indicator. So that would be kind of the ideal welfare indicator we'd be looking for, and there'd be rather few false positive errors and rather few false positive errors in our data. Sorry, just going back, I should credit Georgia Mason with coming up with this graph. I think it's a really useful way of thinking about the value of different welfare indicators and what we should be looking for in a good one. And just to give you a couple of examples of ones that are not so good, perhaps, a welfare indicator I've thought quite a bit about is body weight. And I think this is an example of a poor, very non-specific indicator. So it's non-specific because both very high body weight can be associated with poor welfare and very low body weight can be associated with poor welfare. And there's also quite a wide range of weights in the middle here, where there's not much change in the valence of affective state as body weight changes through really quite a big range. So that's an example of a very non-specific indicator that's probably not going to be very useful for assessing welfare. Another one here is acute glucocorticoid release, which I think is an example of a sensitive but maybe not very specific welfare indicator. So there's evidence to suggest that corticosterone or cortisol levels in blood might increase both with the positive valence of experience, so reflecting excitement, positive excitement, and also negatively valenced experience, so responding to, for example, stress or anxiety that's negative in valence. This has led people to suggest that maybe cort is a better reflector of arousal rather than valence of affective states because of this kind of U-shaped relationship here. So I would argue that this is a very non-specific welfare indicator. Okay, so that's my general introduction about the kinds of features we're looking for in good welfare indicators. I now want to talk a bit more about telomeres as biomarkers of cumulative experience in humans and non-human animals. So what are telomeres? So for those of you that don't know what telomeres are, they are the ends of the chromosomes, and they are present in all eukaryotic cells and therefore all vertebrate species. And they're basically comprised of repeating 6 base pair sequences at the end of all the chromosomes. And their function is to protect the coding portions of the DNA, which are shown here in blue, when cells divide. So when cells divide, a little bit of DNA gets lost from the ends of chromosomes when replication happens. And basically, the telomeres are there to mean that little bit of DNA that's lost is not affecting the important coding portions of the DNA. So typically telomeres shorten with age, especially in proliferating tissues such as blood, for example, because they shorten every time a cell repairs, but they can every time a cell divides, but they can also be repaired by the enzyme telomerase, which an organism can use to build up its telomeres again. So they're somewhat dynamic structures. They don't just get shorter with time. There is potential for them to be repaired. And interestingly, there's substantial evidence that telomere length is sensitive to cumulative stressful experience in humans and animals in a dose-dependent fashion. And I'm going to summarize some of that evidence for you. But this really led to the hypothesis that telomere length is potentially an integrative measure of experience over time. And the nice thing about it is that it's a single objectively measurable indicator. So the idea is that if the experience of a human or animal affects the length of its telomeres, that's a lasting biomarker that we can measure that summarizes something about the experience of that animal. So the balance of telomere loss and telomere repair over time within that individual. So I originally proposed this idea in relation to animal welfare back in 2016, and I wrote a paper in the journal Bioessays about this. And this diagram comes from that paper, and it was my attempt to summarize a lot of the information that we have, mostly from humans, about telomere biology. And what you can see here is I've got 3 columns. So I've got kind of organism level experience, the effects that this has on the cellular environment, and the effects that that has on telomere dynamics. And I've divided my organism level experiences into 2 categories. Ones that I think would be largely associated with negative affective states in humans, and ones that might be largely associated with positive affective states. So obviously this is a bit of a subjective categorization on my part here, and I've had a lot of debates over the years about whether certain things would be positive or negative, and the time span of those experiences. So, for example, diet is quite a problematic one, because we know that eating sugar, for example, can be very effectively positive in the short term, but might actually lead to bad health consequences in the long term. So there are some issues about exactly how we make these associations. But this was what I came up with on my first attempt to do this. So by and large, these kind of negative experiences are associated with higher levels of inflammation in the body, higher stress hormones, more oxidative stress. In general, that's associated with more cell division, less telomerase production, so that's the enzyme that repairs telomeres, and more direct damage to telomeres. So oxidative stress can directly damage telomeres. And so there's an effect on eventual telomere length. So the idea here is that negative experiences predominantly lead to telomere shortening, whereas there's some suggestion that more positive experiences might actually reduce the processes that cause telomere shortening, but also possibly enhance processes that cause telomere lengthening. So there should be kind of opposite effects of positive and negative experience on telomere length. So what's the quantitative evidence for this in humans? So this forest plot here summarizes the results from a meta-analysis that I did with my colleagues Gillian Pepper and Daniel Nettle a few years ago, where we searched the whole of the human telomere literature, and we found 138 different papers where there were different types of exposure that we judged to be associated with some kind of stress in some domain. And there were measurements of telomere length. And what you can see in this slide is that overall there is an association between being exposed to something stressful and having shorter telomeres. So everything to the left of the 0 line here represents shorter telomeres or more telomere shortening being associated with stress. So you can see there's a whole lot of different types of exposures here, from physical disease, poor nutrition, bad environment, psychiatric illness, smoking, alcohol, bad sleep, reduced physical activity, psychosocial stress, poor socioeconomic status. These things are all associated with having shorter telomeres in humans. Interestingly, this sort of association extends to non-human animals too. So I told you that telomeres are present in all eukaryotic cells and therefore all vertebrate species. And this is a similar meta-analysis done by Marion Chatelain and colleagues a few years ago. And they did something similar to our human study, but on non-human vertebrate papers, and they found a total of 109 studies. And again, they find that overall, things that one might conceive of as being stressful or associated with negative affect are generally associated with shorter telomeres in non-human animals. And that paper covers a whole array of different vertebrate species. So it's kind of reassuring to see that these effects from humans are really extending to other non-vertebrate species. Before I go any further, I would like to say a bit about the measurement of telomeres. So you can measure telomeres in different ways. The most common method is what's called quantitative PCR, and that is preferred for a number of reasons due to being requiring a very small amount of DNA, being quite easy to establish in the lab, having a kind of quite low handling time per sample. But it has some disadvantages associated with it too, in that generally, looking at the bottom row of this table, the precision of measurements is much, much lower on average. So qPCR methods for measuring telomeres tend to yield rather poor measurements of telomere length. In contrast, terminal restriction fragment methods, which is a different method for measuring telomere length, which is more difficult to do, requires more DNA, but is actually much more precise, and actually not only gives you a single average measure of telomere length, but gives you a distribution of telomere lengths within the tissue that you're sampling. So there are a kind of pros and cons to different methods, but a lot of telomere research uses qPCR because it's cheaper and easier. And as a result, a lot of it is also, the measurement error is very, very high, and this affects the reliability of telomere measurements. And that should affect your critical appraisal of papers in this literature. So as we saw in the last couple of meta-analyses, the effects are there of stress on telomere length, but they tend to be pretty small effects on average. So we're not talking about large effects here, they're small effects. And I think that's a lot to do with the fact that people are using these very error-prone measurement methods, such as qPCR on average, and that you in fact can get bigger, more reliable effects with this terminal restriction fragment method if you can manage to implement it. Okay, so I want to move on for the last part of my talk now and talk a bit about what I've actually done with this approach in starlings. So you may wonder, why in the world do I work on starlings? I've worked on them for my whole career. They're not obviously a tremendously economically important animal, that nobody keeps them as pets, or very few people. So what's the interest in starlings? Well, I was introduced to them by my PhD supervisor, Alex Koselnik, as a very nice model to use for research in animal decision making. But I've come to realize that they're good for a lot of reasons if we want an animal model to study behavioral and biological problems in. So they're a non-domesticated species, which is interesting in terms of kind of addressing more evolutionary questions because they've not been altered by the process of domestication. Unlike a lot of our laboratory animals, they have a much more human-like life history, I would argue. So they're altricial, they have biparental care, and they're very long-lived compared with your standard laboratory rodents. So a starling can live for up to 20 years in the wild. Usefully, their development is very compressed. So the chicks are in the nest for only around 21 days. And if you're interested in studying effects of developmental conditions on the birds, that's quite a useful thing that their development is so short. They nest in boxes because they're cavity nesters, and this makes them quite easy to work on in the field. You can cross-foster chicks and you can hand-rear them easily. So these are all good things about them. Starlings adapt well to the lab, and we have many cognitive measures available already developed in them, which is useful in terms of trying to validate new biomarkers of effective experience, because we have things like good measures of cognitive bias available to us in starlings. So I worked on starlings in Northumberland, in the northeast of England. We have our nest boxes on farms. You can see some here. You can see inside a nest box here a starling brood when they're about probably 5 or 6 days old, I would say. So just to tell you a little bit about the research that we have done looking at how various types of stress affect telomere length in our starlings. This is an experiment that we did in the field back in 2013 now. So what we did here was to take donor nests and take 4 sibling chicks that were matched for size on day 2 after they hatched. And then we allocated 2 of those chicks to a nest where they were larger than the other chicks in the nest, and 2 of them to a nest where they were smaller than the other chicks in the nest. So our focal chicks were either advantaged or disadvantaged in terms of their size. And the idea with this manipulation was that it would manipulate the stress experienced by the focal chicks in terms of how hard they had to compete with their nestlings to be fed. So the chicks have to compete for food every time the parent comes back to the nest with food. The idea is if you're small, you have to beg that much harder in order to actually be the one that gets fed on a visit. So the focal chicks were in these manipulated nests for a total of 10 days between days 3 and 12. And after that, we took these birds into the lab and reared them under identical conditions. And I should say we took blood samples at the beginning of the manipulation on day 3 and at the end on day 12. So that we could measure how their telomere length changed over that 10 days of the manipulation. So yes, the manipulation was in the field and the birds then came into the lab and we took blood samples at these 2 points here. We then kept the birds in the lab for cognitive and behavioural measures in the following years. I'm not going to say anything about that today. So what did we find in this experiment? Well, during the manipulation, we actually found that growth was identical in our disadvantaged and advantaged chicks. So our manipulation had no effect on their growth rates. The lines are exactly on top of each other here. But interestingly, what we found was that when we measured the telomere lengths on day 12, there was a significant effect of the manipulation, and the disadvantaged chicks that we hypothesized had to compete more for their food and do more begging behavior had significantly shorter telomeres. This was measured with qPCR, and the telomere loss, I'm not showing that graph here, but the telomere loss was also greater. And I should point out that this experiment was in just 37 chicks in the end, so it's quite a small sample size, but it was a really well-controlled experiment in that we had some genetic control by comparing siblings, and it was also done early in development when telomere length is changing most rapidly. And I think these are reasons why we got this very clear result in such a short period of time in this study here. So typically telomeres are going to change most early in development, and that's when they're going to be most sensitive to picking up effects of stress. So I think this is interesting because it suggests that there's quite a subtle effect of this manipulation that we're picking up with the telomere length that we didn't actually pick up with the change in growth in these birds. We followed up that experiment with a lab manipulation where we could actually control exactly what happened to the birds during the manipulation. So this was in the following year. Rather than rearing the chicks in the field with their natural parents, we brought them into the lab when they were only 4 days old, and we fed them rather than having starling parents feed them. And we manipulated both the amount of food the birds got, so either plenty or lean, we called these groups. So the lean group got about 75% of the amount of food as the plenty group. And we manipulated how much begging they had to do. So the chicks were either fed on every visit, or in the hard group, we had 10 extra visits per day when we stimulated the chicks, but didn't feed them. So you can see the experiment here. The chicks are in buckets, and we fed the chicks with a pipette 10 times a day. So you can see some chicks here begging away and being fed in a minute by one of the group with a pipette, so that we could control exactly how much food each of the chicks got. So there you can see the chicks being fed with the pipette. Okay, so whoops. So sorry. So yeah, the idea here was to manipulate two different components of stressful experience. The amount of food, so something to do with nutrition, and the amount of begging that the chicks had to do, and look at how these contributed to changes in telomere length. And our prediction was that the lean hard chicks, the ones that got both little food and had to do more begging, that would simulate perhaps the experience of our disadvantaged chicks in the field in the last experiment. What you can see is that there were some differences in growth rate here. So if we look at the plenty chicks, the blue ones that had ad lib food, there was actually no difference in how fast the plenty easies and plenty hards grew. So perhaps they're a good comparison with our field experiment. The lean chicks did actually, on average, grow slower, although they caught up after the manipulation, with the lean hards growing the slowest of all. In terms of telomere loss, so this is the change in telomere length between day 5 and day 56, which was actually after the birds had had a chance to recover after the manipulation. You can see that we have a really interesting pattern here, that telomere change is greatest and most negative in the lean hard group, and least in the plenty easy group. So the treatments go in exactly the order you would predict, that there's more loss in lean hard and less loss in plenty easy. There's actually no interaction in this study, so it's an additive effect of the two kinds of stress. But what we found was that the begging, the work, had the larger effect on telomere loss. So this sort of reflects what we were seeing in the meta-analyses that I showed you, that it looks like different kinds of stress can both affect telomere length, and that they can actually add together in this case in an additive way to affect the total amount of telomere loss that you're seeing in these chicks. Okay. So just one final starling study here. So the two I've just told you about were in very young birds, and when they were still in the nest. This is a study that was done for a much longer term period, so 19 weeks in the lab with adult starlings. So here we had a between subjects design where birds were either in a control group where they were fed ad libitum, or an insecure group where their food was removed for a random 5 hours unpredictably on 5 days of the week. So this was designed to simulate what we call food insecurity, so not knowing when you're going to have food available and when you're not. And that went on for 19 weeks. And what we did was caught and weighed the birds every couple of weeks for 19 weeks. And we also took regular blood samples to measure their telomere length. what you can see here is that this was over the winter and there were some changes in body mass, but from the start of the manipulation, the insecure birds were always fatter, and that was a predictable response to unpredictable food in the starlings. It's been documented many times that when you make their food unpredictable, they tend to put on weight. And interestingly, that putting on of weight was associated with shorter telomere length in the insecure birds. So what we think is actually going on here is that the birds were diverting energy from maintaining their bodies, so maintaining their telomere lengths, into putting on extra fat to ensure themselves against risks of starvation. So there's a trade-off going on here, that the birds are kind of reducing their investment in somatic maintenance and repair, and that's reflected in shorter telomeres over this study. In this study, we use the TRF method, so we've got absolute telomere length measures here. The results, the effects were quite small, and we needed this whole 19-week period to get something significant actually coming out. And in fact, it was only in the longest telomeres that we saw these effects, so it was a much smaller effect. I think reflecting the fact that telomeres are less sensitive to stress typically in adult animals when they're not growing so rapidly. All right, so just to do a bit of summarizing to wrap up what I've said to you, I've argued that we need reliable and valid indicators that can be objectively measured to infer the welfare of non-human animals. And I've tried to show you some evidence that telomere length might have features for doing this. So I've shown you that there are huge amounts of data in humans that we can use for validation purposes, where we know that there are negative or positive affective states associated with changes in telomere length. There are also growing amounts of data from non-human animals. So I've shown you the Chatelain meta-analysis and also some of my own experiments that I think show very clearly that you can manipulate telomere length by manipulating the stress that starlings are exposed to. An advantage of telomere length is that the sample collection is fast and relatively non-invasive. You only need a tiny amount of blood, particularly for qPCR, a drop of blood from a bird, which have nucleated red blood cells is plenty to measure telomere length. You'll need more in mammals where the red blood cells don't have DNA in them, but you can still measure it from the white blood cells. You'll just need slightly more blood. So relatively fast and non-invasive sample collection. We've seen evidence that telomere length is decreased by negatively valenced experiences. And there's also evidence for it being increased by potentially increased by positive experiences, although I'd say the evidence for that is much less strong. And we don't have any in animals at the moment, only in humans. So that's an area that really needs to be addressed in animals, the association between positive welfare and telomere length. I'd say that there is evidence for dose dependence. So more stress causes greater telomere shortening. We saw that in our starlings, different kinds of stress can be integrated in telomere length. So increased begging and reduced food both led to decreases in telomere length in our birds. So yeah, combining different kinds of stress, so poor diet and more begging behaviour. So altogether, I'd say that this suggests that telomere length is a good candidate biomarker for cumulative affective experience of animals that might actually be quite useful in situations where we want to sample welfare on a large scale, such, for example, in farms or maybe at slaughterhouses at the ends of animals' lives. We might want to use it to give us a kind of integrative measure over the whole life of an animal towards the end of its life. There are, however, a number of limitations of telomere length. So although I've said that blood is relatively non-invasive, it's still potentially a stressor to the animal. So taking blood samples may not be okay for everyone. other tissue samples are possible. So you can use um, cheek cells or potentially cells from milk. That's been done. So there are other options, but you need to have good quality DNA, and ideally it needs to come from proliferating tissues where telomeres are going to be changing in length. So there are some issues, and most of the validation data we have is from blood. So ideally blood is what you need. Measurement error is a real problem. So I just touched on the fact that there are different measurement techniques, and they can be done either well or less well. But typically, the cheaper, easier methods are more error prone, and that's a real issue. There are also large individual differences in telomere length, and the implications of that are that longitudinal studies will be most powerful because it's going to be better if you can use each animal as its own control and actually look at changes over time. If you're just looking cross-sectionally using a single telomere measurement, that will often be very problematic because there's such big individual differences. You'll have a lot of error, which will mean you'll need huge sample sizes to actually see effects. Another thing I've touched on is that telomere length is going to be most sensitive to experience in young animals. This is because there's a lot of cell division going on then, and telomeres are changing in length quite a bit. And there seems to be greater sensitivity to stress during that period. In adults, you're going to be most likely to see effects if you measure them over really long periods of time. So in humans, people typically think of 10 years as being a good period to assess effects of experience on telomere length for. So that's something worth considering. And telomere length is unlikely to be useful for very short-term measures of stress in adult animals. And I'd say there are some potential problems with specificity. So some experiences that are effectively positive in the short term, at least, like, for example, eating sugar, which would be regarded as poor diet in humans, may actually shorten telomeres. So I think something to think about here is that telomere length may be a better measure of cumulative health rather than cumulative effective state. In many cases, I would argue that these two things will actually overlap, but there may be cases where they will actually depart, and we need to think quite hard about that and the tensions between kind of health-focused accounts of welfare versus effective state accounts. So just to summarize all that on my graph, I do think there is good evidence for a monotonic linear association between telomere length and effective experience. There might well be errors due to insensitivity to short-term experiences, and there might be false negative errors due to measurement error and big between subject variability in telomere length. There might be some false positive errors due to measurement error, and also effectively positive but unhealthy behaviours, as I've suggested. So I think it's a good candidate, but there are some limitations that you need to be aware of. So I'll finish there, and I'd just like to say thank you to various people who've influenced the ideas in this talk. Paul Martin, with whom I wrote the new edition of Measuring Behaviour and influenced my thinking around validity and reliability of measures, and Georgia Mason, who's really a wonderful friend and colleague who's challenged me to think hard about the validation of welfare indicators over the past couple of years. And many of the ideas in this presentation actually come from Georgia, and you should look out for her upcoming book on valid welfare indicators, which she's co-editing with Mike Mendel. And I should also thank the Wild Animal Initiative, who are actually funding some of my current research on starlings. So thank you very much for listening, and I'm very happy to take any questions at this point.
Thank you so much. So I have some questions in the chat, but before I start taking questions from the chat, maybe I just want to ask you about the issue of begging behavior in the starlings. I was wondering, how do you know which of them? Because when I looked at the picture of the chicks in the bucket, how would you know which one is begging more, or how do you know this one is begging more? How do you analyze that? How do you get?
So are you talking about our hand rearing experiment in the lab?
the begging behavior of the chicks. How do you know which of them is uh displaying more begging behavior? Is it in terms of vocalization or what?
So we didn't measure the begging behaviour. We didn't measure it in the field at all. We just assumed that it would be the case, and that's why we went on to do the lab study. We didn't measure begging in the lab, but we manipulated it. So our easy, hard manipulation was how much begging we made the chicks do over the course of the day. So in the easy groups, we would open the bucket and feed all the chicks in the bucket and then put the lid back on. In the hard manipulation, 10 visits were like that. But in another 10 visits, we would take the lid off the bucket and just stimulate all the chicks to beg with the pipette, but not actually feed them at all for a similar amount of time as a feeding visit. So they got about double the amount of begging overall compared to the easy group. So we were basically just doubling the begging that all the chicks in the bucket were doing in that experimental design. And then that was cross factored with manipulating how much we fed them when we fed them. So the plenty ones got fed ad libitum on each visit, and the lean ones were fed 75% of what the plenties got. So that was how the manipulation worked. We didn't measure the begging, we manipulated it in that study. And the assumption was that doing all of this very, you saw the behavior, it was quite vigorous begging behavior, that would be stressful in some respect for the birds.
Thank you so much for showing that. Also, or still on that experiment, how were you able to maintain like aeration, ventilation? Because I saw them in the bucket and it was completely covered. So how do they breathe? What did you put in place to sort that out?
So I mean, the idea with the buckets was they were to sort of simulate the somewhat like the natural nests in the wild, which are these nest boxes that I showed you. So these birds nest in cavities, so they don't have a lot of natural ventilation in the wild. They're often quite enclosed spaces, so maybe in holes in walls or trees. We And they're dark typically, so we had just a cardboard lid on the buckets to keep it dark, but there was sort of some gaps around, so there was no issues with air getting into the bucket. So the idea was just to keep them dark and somewhat warm, but to allow the air to come in around the edges, so they weren't completely sealed off.
Thank you so much. So I go into the chat, and there is a question from Megan Barrett from Indiana University, Indianapolis. She's asking, does relying on humans as a gold standard limit us to only affective states we assume are shared between the species of interest? Is there
Yeah.
okay.
Yeah, I mean that's a good question. I mean we have lots of different methods for validation open to us, and I just say that's one of the methods that I wanted to draw attention to. I mean, because with telomeres we have so much data from humans. I guess knowing what kind of changes in biomarkers are associated with negative affective states in humans is a good starting point for related species. Obviously, as you get more and more phylogenetically distant, I think maybe there's an issue with whether or not these things will translate to other species, and that's something we need to think hard about. I think having these within species experiments where you manipulate something that you're pretty sure will cause negative welfare is really important in more distantly related species. So it's a question of how you know what that is. I think that is a real difficulty that we face, so as not to kind of get involved in kind of circular reasoning about how we're validating our indicators. But I think I mean things like poor health seem fairly it would be hard to argue that would be a pleasant experience for most animals. So being deprived of enough food or having to work super hard to get the food you get, these things, I mean, our intuition in starlings is that these would be associated with poorer welfare. But it is a problem. I mean, there's not a simple way around this. We just need to use a number of different methods and hope that they will provide convergent information, I think. There are other ways of doing it too. So you can use drugs that we know have effects on affective state in humans. So that's been used a lot in the context of pain, and that seems to work well even in very distantly related animals such as invertebrates. So yeah, validation is a real problem that we need to think hard about.
Thank you, Melissa. There's a question from Marisol. Thank you, Melissa. Fascinating. Which cells did you use to measure the telomere length? Is the age of the cells affecting their length, old versus young cells?
So in our Starling studies, we took blood samples and our telomere length measures will predominantly have been from the red blood cells in those samples. So the red blood cells are the vast majority of the cells in blood. And because birds have nucleated red blood cells, you can get DNA from all of their red blood cells. So a really good reason for doing telomere work in birds is that you're using a very homogeneous population of red blood cells to get your telomere measurements. In mammals, where you've not got nucleated red blood cells, usually it's the white blood cells that are used from the blood, and these are a much more heterogeneous population because there's different kinds of white blood cells, and some of them will proliferate more when you have infections, for example. So that really complicates interpretation of telomere data in mammals, that you can have, what can look like changes in telomere length can actually be due to changes in the proportions of different cell types in the blood. So that causes some complications. But you need to sort of think hard about what it is you're actually measuring. Does that answer your question? It's going to be different in different species. And I think, yeah, birds have been a good model for this research because of the fact that they have this really homogeneous population of cells that we could access easily.
Okay.
And also blood turns over fast. So telomere length is changing in blood faster than, for example, in a tissue like muscle, where it barely changes at all over the lifetime.
Thank you so much. So will you say using telomere length as an indicator of welfare, is it good? Will it show us, does it work for short-term stress or more of a chronic term stress?
I would definitely say much more for chronic stress. So most of in the human literature, the sort of recommendation is generally that you would look for effects over really long periods, like I said, 10 years. So for example, attempts to look at effects of damaging behaviors like smoking on telomere length, and typically they're collecting data over 8 or 10 years to see an effect. And that's because telomeres are changing very slowly in adult humans, so there's less sort of potential for you to see effects there. So there's a sort of you can get away with shorter periods if the animals are really young. So as you saw in our starlings, we could detect quite subtle effects of stress in just 10 days when the birds are in the early stages of development. So when they're growing really fast, I mean, they basically go from 5 grams to 80 grams in less than 2 weeks. So they're going to their adult weight in about 2 weeks. So there's masses of growth going on there, masses of cell division. And I think that's why we could see these big effects in these animals. Whereas in an adult animal, like the adult starlings, the effects were much, much smaller. And that's because there's not nearly so much cell division going on. And so there's less potential to see these effects of stress on telomeres. So in general, I think it's absolutely a measure that you should use for looking at chronic effects of different kinds of experience. So the type of housing an animal's in, chronic disease, chronic deficiencies in diet, or environment of some type. You shouldn't expect to be able to use it to look at really, really acute effects of, one stressful experience like having a procedure done on you or something.
Yeah, thank you so much for that. So I go back to the chat. There's a question from Samuel Joshua, also from Indianapolis. Yeah, how cheap is the use of telomere length in the assessment of animal welfare? For example, how much dollar or pounds will be the cost, to do this procedure?
That completely depends on what methodology you use. So qPCR is pretty cheap, but you need to make sure that you're really, really checking your measurements are repeatable and good, and that you've got good methods there, because it can yield, in my experience, total rubbish if it's not done very carefully. The TRF methods are much more difficult to set up and more expensive and more time consuming, and will cost a lot more. I can't give you exact numbers. I haven't done any measurements myself recently. I've always collaborated with other people, but yeah, it really depends on what method you choose. And there are other, I've only mentioned two methods there. There are others too that are, will give you even more information and are more complex, but have not really been used in animal science contexts at all so far. So there's a range of methods that cost different amounts, and have require really different expertise as well.
Okay. Yeah, you talked about, there's just a follow-up question here that from measuring the telomere length from taking blood samples and also tissues, is there any study that has validated whether you get this the same result using these different samples, blood
Yes,
samples, tissue samples?
yeah. So there's quite a few studies like that. So in general, I think the finding seems to be that you get correlations between changes in telomere length in different tissues. So, for example, I know of one paper in humans that looked at changes in blood and changes in muscle, and they are correlated within individuals, but the changes in muscle are just tiny compared with the changes in blood. So, you're going to get bigger changes in proliferating tissues where there's lots of cell division going on typically, and much less changes in tissues where there's very little cell division, like for example muscle. But there are a number of studies suggesting that telomere length changes in telomere length are correlated across tissues. And there are studies validating, for example, measurement of telomeres in unconventional tissues like swabs from the mouth, so cheek cells, which is often used in children and has been used in dogs as well. So there's some nice studies where they don't want to take blood samples where they have validated use of cheek cells, but those cells are quite problematic because there tends to be a lot of dead cells with bad quality DNA. So I think there are people that think that those cells are not the greatest ones to use. I know there's at least one study using milk in cows, which is an interesting one. I think that's very, very plausible. And I think that's been validated. Um, and you can use things like, I think with bats, they've used little biopsies from their wing membranes, for example, or tips of tails from meerkats. So there's a range of different methods that are used. I'd say you probably need to validate them against a more accepted measure, which would usually be blood, ideally.
Yeah, thank you so much. There's a question here from Sanusi. He wants to know if you have considered telomere length measurement stability over time.
So by that, do you mean measuring the same individuals repeatedly over time? Um,
I think so.
yeah, I mean, I didn't show you any data, but it did do this in our Starling studies. I only told you about the changes over the developmental manipulation. We did do follow-up measures in some of our studies a year later. And in the Starlings that had the developmental manipulation, the relative lengths of their telomeres at the end of the manipulation at like day 12 or whatever it was, predicted the relative telomere lengths a year later when they were a year old. So the sort of ranking of the birds at the end of the manipulation was sort of somewhat maintained a year later. So those differences were somewhat stable over time. So what happened early in development was kind of still reflected in the telomeres of the birds after a year. So there is data on that kind of thing, yes.
There are lots of people, appreciating the talk. Thank you so much, everybody, for joining us. I still have my own questions here. Um, okay. I was one when you were talking about an experiment where you compared the body weight of chicks that were subjected to unpredictable feeding and the unpredictable feeding chicks. Yeah. And you reported that they had more body weight. They put on more body weight gain because of this unpredictability in terms of food. But I was now wondering, you explained the physiological basis, which I understand, but I want to know if there are some behavioral observations that you noted. Maybe they were less active, or something like that. What behavioral change did you notice?
So, so I didn't really have time to give you much of the background to those experiments. The experiment you're referring to was the one we did in the adult starlings, not in the chicks. So the long-term study where we were looking at the effects of taking away their food unpredictably. I have done a number of similar related studies to try and understand how unpredictable food affects animals. That's the only one in which we measured telomere length. But yeah, the interest in telomere length partly relates to how the birds are managing to put on weight despite the fact they're actually getting less to eat. So we've many times we've documented this phenomenon that you make their food unpredictable by taking it away for periods of time. And what happens is the birds gain body weight, which we interpret as an adaptive response to ensuring themselves against periods of, lack of food availability. Um, but the interesting thing is they do that despite the fact that they eat less overall. So it's not that they compensate by eating masses when they get food back again. They don't do that. They eat less overall. So that raised the question of where is the energy coming from? They're putting on fat and yet they're actually taking in fewer calories. So you're right that that means they have to be spending less energy. And the the vast majority of the energy we spend is actually on our basal metabolic rate, which is in us and other animals, which is maintaining the basic functioning of the body, and obviously maintaining your cells and your telomeres is part of that. So my argument was that these animals that are putting on body fat are doing it by taking energy away from maintaining their telomeres and diverting it into fat stores that allow them to survive in the short term. Um, so that's an interesting piece of information about how energy is being reallocated. So you're right that there could be behavioral changes too, because part of your energy expenditure is in physical activity. So we did look at whether there were changes in behavior as well in these birds, not in that experiment, but in different ones. And we've got inconsistent results there. So there is some evidence to suggest that they're less active. So we did in some studies see the birds were spending more time roosting inactive when they were in the unpredictable food conditions. So there might be what I think is there are a whole number of mechanisms they're using to reduce their energy expenditure. They're doing less behavior, so resting more. But they're also spending less energy on maintaining their bodies and their cells. So we also recorded their feather growth, for example, and we found that they regrew. If we plucked their feathers out, they regrew them more slowly. So that was another bit of evidence that they're investing less in repairing their bodies, repairing their telomeres less, repairing their feathers less, and diverting that saved energy into growing their fat stores. So that's quite interesting biology about what's going on there.
Mm. That's interesting. I was just wondering, now that you are focusing on the use of telomere length in as a welfare indicator on a long-term basis, I'm also aware of Tom Smilder, who does things on adult hippocampal neurogenesis, and I was wondering if both of you have collaborated to see if these two biomarkers actually, point to the same thing.
No, I mean, Tom's a very good colleague of mine, and we haven't ever looked at both biomarkers in the same animals for a number of reasons. I think I have collected brains from some of my starlings at the end of these studies, and they're still in the freezer waiting for somebody to look at their brains. Actually, measuring the biomarkers of hippocampal neurogenesis is really hard and time consuming. The way Tom's doing it, involves a lot of counting cells down the microscope. So it's not a quick thing to do. There are some studies in humans that have looked at correlations between telomere length and hippocampal volume using not histology, obviously, but MRI, so structural MRI to get estimates of the size of the hippocampus from MRI. And those studies do seem to suggest that there are correlations between these two biomarkers, which makes sense because they're both measures of biological age. And so we've speculated that they should be correlated. But yeah, I've never done both in the same animals because the hippocampal stuff is hard to do.
Yeah, understand, yeah. And one more question for me. Before I take that question to the audience, if you have any other comment for our speaker, feel free to use the react button to let me know, then I'll unmute you. And also feel free to turn on your camera if you want to come on camera. And yes, we'll be having like 10 minutes of interaction. And yeah, we need to speak, discuss. If you have anything related to this topic, feel free to let me know and I will unmute you to talk about it. So the final question for me here is, now, is there any correlation between a reduction in telomere length and longevity?
Yes. So there are data on this in both humans and in birds. So probably in humans, one of the nicest studies was a study of identical twins, where they measured the telomere length of identical twins, and on average, the twin with the shorter telomeres was the one that died earlier. So that's kind of an interesting human study. In birds, there's really nice data showing that telomere length predicts longevity in a few species. So yes, I mean it's a good biomarker of biological age, and it predicts longevity in the studies where this has been looked at on average. I mean, I don't think it's going to be very useful at the individual level because of all the problems with measurement error and individual differences in telomere length, but on average, in population studies, you do see these effects.
Oh, thank you so much. Yeah. So to the participants, it's time for you. Let me know if you have any further comment for our presenter. Kindly use the react button. Melissa, can I ask you this question? For especially those developing countries that have little funding to carry out some of these, procedures, what do you think is the future area that one can also look into as, welfare indicators that might be easy to assess or to conduct? Yeah.
Well, that's a good question. I suppose, I mean, I try to give you a sense of what I feel like the advantages and limitations of telomeres are. And I mean, I think a real limitation is that they require having the lab and the expertise to do the measurements, and that's obviously, not everybody's going to have that available to them. So maybe in situations where there's no funding for that sort of thing, it's just not a method that's feasible. And we need to focus on things that are cheaper to measure, like behavior, for example. I mean, I think a lot of behavioral measures are really, really good. They're just difficult to do on a very large scale, for example, in farm situations. So I think part of the attraction of the idea of telomeres was that it could summarize the whole lifetime experience of an animal, possibly at slaughter. You could take a single blood sample before an animal's slaughtered, and that might give you an indication of, the relative conditions on different farms, for example. So you might be able to actually, at the population level, compare different housing conditions or something, by looking at the impacts on telomeres over animals' whole lives. So that's the sort of attraction of the idea, that it's actually feasible to do on a massive scale if you've got the funding, and it can capture all of these different sorts of experiences. But I mean, I think that there are behavioural measures that are also really, really nice that can be done with, a pencil and paper and a watch or something, that don't require PCR machines or other technology in the lab. So I wouldn't want to put people off doing simple behavior-based welfare assessments. I think they've got their place. And actually, telomeres are by no means perfect. There's just a lot of measurement error, and you could spend a lot of money and get useless data. So, I don't think it's the be all and end all at all. In fact, I've kind of become a bit more skeptical about it as I've got older and seen some bad data sets as well as some good ones where people have invested a lot of time and money and got nothing. So, I think it's something that is potentially interesting and good, and has particularly interesting characteristics, like allowing this kind of integration of many different forms of stress over time, but also has some real methodological issues associated with it. So, I think we should be having a number of different measures, and trying to always measure more than one thing, because there's not going to be one indicator that ticks all the boxes. And we'll get a better assessment of animal welfare if we do triangulation and measure lots of different things and see if they all converge on the same answers to our questions.
Yeah. Yeah, thank you so much, yeah, Melissa. So I was just wondering if you have the final words for us.
I think my final word is, well done on getting this seminar series going, and good luck for what you're trying to do. I think it's really positive, to bring the message of animal welfare research, to the whole world, and to the global south in particular.